Oxford. Now, this area of land they knew had ticks on, and they'd seen some tick-borne diseases in the herd in previous years. And so they decided to import a red water vaccine, so a Babesia vaccine, that had been developed in France, and vaccinated the stock with this vaccine before turnout. They turned out the stock in early February 2019, and 3 weeks later, trouble began, and the vet gave us a ring. So the farmer was reporting that 6 out of the 20 adult cows were presenting with illness. They were reporting clinical signs of the cows lying down and being dull. And when they went to examine them, the mucous membranes appeared either pale or in some cases yellow, and really high numbers of ticks were seen on the animals. And 4 out of 20 of the cows died. And this presentation was more severe than they'd seen in previous years due to tick-borne diseases, and so they were a little bit concerned and wanted to investigate it further. So we had 1 adult cow into APHA star cross, and we performed a post-mortem examination on it. Now, as Bev said, sometimes you don't see very much with tick-borne diseases, but in this case we found that the cow had a very enlarged spleen, as you can see that big red bit in the picture. The mucous membranes, so the conjunctiva, were really very pale, and there was red urine in the bladder. And for anyone who's seen red water fever in cattle before, you know that red urine really is quite characteristic of the disease in the later stages. So following the post-mortem findings, we wanted to do a bit more diagnostic testing to investigate this further. Now, at APHA, we have what's called a pan-pyroplasm PCR, and this is a test that's currently in development. So you won't find it on the commercial price list at the moment, but it can be very effective for diagnostic investigations such as this one. Now, the beauty of the test is that you can test a whole range of samples. So you can test blood samples from either live or dead animals, and you can also test tissue samples such as spleen, and you can test ticks. And for those of you that aren't as familiar with PCR tests, basically what we're aiming to do is we extract genetic material from our samples, and then this genetic material undergoes a series of kind of diagnostic processes until we basically are left with a genetic barcode. And this barcode we can then compare against known barcodes of blood pyroplasms in this case. And so this test can pick up multiple different blood pyroplasms at once, as I'm about to demonstrate. So the blood sample from the dead cow plus 3 blood samples from live cows were PCR positive for Babesia divergens, which is the causative agent of red water fever. Again, the blood sample from the dead cow plus blood samples from 2 others on the farm that were alive were PCR positive for anaplasma, so the causative agent of tick-borne fever. And therefore, we confirmed co-infection in 3 animals that were affected on this farm. As I said, we can also test ticks, and it's slightly gross, but they basically pull the legs off the adult ticks to put through the test. And so 3 of the 10 adult ticks we collected were positive for Babesia divergens, and 2 further ticks were positive for anaplasma. And it's worth saying that we couldn't find co-infection in any ticks, so these are separate ticks that are positive for Babesia and anaplasma. The other thing that we did, as Jolyon mentioned earlier, was we sent some ticks away to the Public Health England surveillance scheme, and they came back as Ixodes ricinus species, which is what we were expecting, which is the classic tick that we see here in the southwest. So why did this outbreak happen? It was unusual to see a case of Babesia in February. Generally, we see cases of Babesia when the ticks are most active, so that's often between about April and July. And as I said, February is quite unusual to see this. So we started having a look at the temperatures for the time of this outbreak. And if you can look at the graph on the right, the dotted lines are the average temperatures, and the solid line is the actual temperatures for when this outbreak occurred. And you can see that for a lot of February, the actual temperature was above the average high. And as others have said, the life cycle of ticks is really complex, and it relies on a lot of different environmental factors, including humidity, temperature, and availability of hosts and the habitat. So we think that potentially this higher than average February temperature meant that the ticks were more active earlier in the season than we would normally expect them to be. And so when these cattle were turned out in early February, they were met with a high tick challenge immediately. And so they were just overcome with the pressure of infection. We don't know what effect the vaccine had. So because the vaccine's not produced commercially in the UK and it is a research-based vaccine that's been developed in France, we don't have the efficacy studies to know how effective it is. So we just don't know whether the vaccine played any role in this outbreak or not. We also think that the co-infection of Babesia and tick-borne fever potentially led to an increased severity of disease. So we know that in other countries, there's been a paper in Sweden, for example, where co-infection of tick-borne diseases in cattle has led to more severe disease. But the reasons behind this are quite complex and we don't really understand them properly at the moment. And I would urge that if you're seeing, unusual outbreaks of tick-borne diseases or suspect tick-borne diseases in your own stock, then always consider that there may be more than one tick-borne disease present and investigate it fully. So this farmer chose to treat the affected animals that were still alive with imazole and with topical tick treatments. And we came up with some control measures for the future. So as was mentioned earlier, calves basically develop an immunity without showing clinical signs to Babesia if they're grazed with infected ticks before about 9 months of age. So that's one point. This grazing needs to be strategic. So we know that there's also tick-borne fever in the tick population. And so there needs to be a strategic use of topical treatments and strategic exposure to ticks at a young age. We also advised that the farmer continue to try and detect cases early and keep a close eye on these cattle and to treat clinical cases to prevent them from dying. And again, it's really important to keep monitoring what diseases are occurring on farm. So I would urge anyone that's seeing ticks on livestock to repeatedly, over each season, send ticks to the Public Health England tick surveillance scheme, not only to contribute to the UK surveillance on ticks, but to also know what's going on your own farms. And that's it. So I'd like to thank Paul and Nick at APHA Weybridge. And this is a really good example of when we call on different people for expertise, because they're really knowledgeable guys at Weybridge that can offer lots of advice on these type of cases. I'd also like to thank the private vet and the farmer involved with the case for their cooperation. And for anyone interested, this case has been written up in Parasites and Vectors, if anyone wants to read about it further. Thank you.